Okay, so this is where we had left off last time, that we had just started to talk about the microbial community, and this was a case study which I was trying to present to you regarding anaerobic digestion process, whereby the solid material which is actually left after harvesting the corn, that is the corn, straw, stem, leaves, etc., they can be used as a source of energy, and methane can be produced by fermenting them. So I had just given you in slide 9 very quickly. I will remind you that these are essentially the anaerobic digestion process was divided into 4 phases. The first phase was the hydrolysis of the biopolymers, etc. And phase 2, which is called as the acidogenesis, is formation of volatile fatty acids, which are short chain fatty acids. And the third phase is called as acetogenesis, that is production of acetic acid, which is eventually converted into methane, which can be used as a source of energy. So now in this reactor, the objective was that all these 4 phases are divided now into 2 phases. The first phase, which is shown here, is the one. So this is the reactor vessel in which both hydrolysis as well as acidogenesis is performed. And for the reasons which are cited in the paper, the objective of the paper was to be able to separate different phases. Okay, so the first phase of hydrolysis and acetogenesis is performed here in this vessel, whereas where you see as the second phase reactor, so acetogenesis as well as methane formation was carried out in the second reactor. Now there is a modification. The way the substrate is fed is what is called as it's a leachate system, where the solids are actually collected in here because you are dealing with a solid substrate, and water is allowed to percolate through this. So the idea is that anything which is soluble, particularly the sugars, the proteins, and many of the fatty acids, are extracted out, and these are all collected at the bottom. So now you have something, you have like a slurry, but it is free. It is only what can be called as the percolate or the leachate, and that is collected into the percolate vessel. And similarly, there were other 3 reactors which are lined in parallel, and the function here is only to be able to extract the percolate from the solid substrate. All of these are stored temporarily in a percolate vessel, and eventually it is fed into the first reactor. Okay, so is this clear? That is what is the layout of the bioreactor here. Is this clear to everybody?
Yes, ma'am.
Okay, so now our objective
Yes,
in
ma'am.
this study, okay, so our objective is here to carry out a metabolic analysis, that is what kind of reactions, chemical reactions are going on here as a result of microbial activity. So what we are going to do here is to collect samples at regular intervals from the first phase reactor and do, like it says in the paper, a metabolic dynamic analysis. That is, what kind of metabolites, what kind of gases are being produced here. At the same time, you also want to monitor what happens to the microbial community as a function of time. Okay, and eventually what you want to do is the output in this reactor, which is formation of fatty acids or formation of gases, etc. An attempt will be made to correlate the production and the process outcome with the microbial community distribution. Okay, that is our long-term objective. So what it gives you is that it gives you a handle on monitoring the performance of a bioreactor. And if there is a need, one can also bring about a change, because in an ideal reactor, when you are getting very good methane formation, there is a certain microbial community distribution. So as you monitor the microbial community dynamics in your reactor, you are able to monitor the reactor performance. And if you feel that there is a certain community which is going down, as a result of which methane formation is also going to suffer, is it possible for you to make an intervention at that stage, at that particular inoculum, so that the dynamics can be shifted in a direction where the reactor performance is going to be increased? So now let's look at the data from the phase 1. So what you will see in this data is panel A. And what you are looking at here, for example, if you see on the y-axis, you are looking at organic acid concentration, which is measured in gram per liter. So it is a sample which has been removed. On the x-axis, you are looking at the days, and the samples are being removed from the phase 1 reactor, where you have acidogenesis as well as hydrolysis, which is occurring. So you will recall that when we discuss the biochemistry, you want to look at, for example, the production of volatile fatty acids, which can all be monitored. The samples are removed, and then the solids can be centrifuged out. The clear supernatant now, which contains the acid, can be injected into an HPLC system. You also prepare a standard with lactic acid or with different kind of acids. So these are all shown here. If you see, like in this figure, for example, the black dot here refers to acetic acid production. So axis you have dictation on do that because, well, with the standard, you have a standard graph. So depending on what you are monitoring, you know how many grams per liter of that particular acid is being made. So in black, we are looking at the profile of acetic acid. In red, like you see here, for example, you are looking at the profile of propionic acid, butyric acid, caproic, lactic acid, and hydrogen. So hydrogen is a gas. So right here, you will see, for example, is that you will have gas, and in blue, you have lactic acid right here. So is this clear to everybody as to how to interpret the data?
Ma'am, the minimum or
Yeah.
maximum, does it interpret something?
Sorry, what is the question?
Ma'am, the minimum and maximum of any of the acid, does it imply something?
Obviously, see, because first I want to know whether the general layout of the experimental results is clear to everybody. Okay, now what you see here is that the entire phase during the first, because the time that you are looking at 110, if you see down here, the time is in days. So over a period of 110 days that you are monitoring the fermenter performance, there are two distinct phases which have been listed here. One is the G phase and the other is the L phase. All right, so there is a clear subdivision into what is called as a G phase and the L phase. G phase is the gas production, which is actually the hydrogen gas which is being formed. Okay, L is the lactic acid production. So what you see is that there is a distinct cycling of the gas phase. So initially there is high production of gas, which is the hydrogen gas, which is followed by lowering. And then again, it goes high, it lowers down, and then it again goes up. Similarly, if you were to follow the lactic acid production between the 20th and the 40, between the 20th and let's say this is like a 35th day, there is an increase in the lactic acid production, which goes low, so on and so forth. So there is a cyclic nature. So the entire fermentation profile, all right, can be divided in the first phase of the reactor into distinct gas phase and the lactic acid phase. Okay, now at this time, we are not trying to make a prediction that this is the way the system, you're only trying to monitor. You're only trying to look at what are the process parameters, and from there you are going to go back and explain the biochemistry of the reaction. But what emerges out in the first phase is that there are two distinct phases. The gray ones are the gas phase where hydrogen production is the maximum, and blue is the lactic acid production phase. In addition to that, you also see that apart from, for example, propionic acid, which shows a very high level of production in the initial phases, but eventually there is a little cyclic nature, but it is not as much as the lactic acid production. But yes, a large number of these volatile short chain fatty acids are being produced during the entire phase. Now in the paper, if you will from the second phase, a part of the reaction mixture was drawn out from the second phase during the first 110 days, and it was recycled back because this is a reactor which operates with cell retention principle or solid phase retention principle. So the contents of the second phase were also recirculated back. But what is interesting to note here is that in spite of that recycling, during the first 110 days, there is no methane formation in the reactor. The only gas which you detected was hydrogen. Okay, so that is what I want you to understand right now, that that this is the way. If you were to monitor the performance of the reactor, this is the way there is distribution of various metabolites. Now, in this case, for example, there is other process parameters are monitored, such as the pH value, for example. So during the time, what is the pH value? And as as is normal, you will see that the pH values are pretty low. Okay, so the pH values, like you see, for example, in the lactic acid production phase, are much lower. It's around 1.5 or 2, and that is to be expected because lactic acid is going to acidify the reaction. So during this phase, in comparison to the gaseous phase, the pH in the L phase is quite low. That is expected. All right. And specific gas production is how much of hydrogen gas. There is no methane. So specific gas production is the hydrogen which is being produced per ml of the hydrogen gas which is produced per total solids per day. So that is what we mean by specific gas production per gram of the solids which are contained in there, which are fed into the bioreactor. And what you see as crosses here are the time point of feeding, because there is a continuous, there is not a continuous, but it's a semi-continuous feeding. So after some time, the percolate, for example, is recirculated back, all right, and fresh substrate is being added, and that is what is shown by a cross here. Okay, so is this part clear to everybody as to what is the way in which the data should be presented?
Ma'am,
Now,
G and L are the gas and the liquid phases, na?
no, not the liquid phase. L refers to lactic acid production. Because you see this blue line, if you just go here and try to understand, that blue here is for lactic acid. So L here doesn't mean the liquid phase. L means lactic acid production. So during this part, which is between 20th and 35th day, and between this phase, for example, there is an increase in the lactic acid production. And that is why this phase is labeled as the L phase.
Yes, ma'am. And G is the gas phase.
G is the gas phase, yeah, because you see this light brown color here, and you go back to understand what is the symbol refer to. It refers to hydrogen production. I hope you can see the movement of the mouse here, because this is your hydrogen.
Yes.
Okay, so now very quickly, now what is interesting is that, there is a lot of monitoring which you are doing with respect to the gases and the lactic acid production. But now you also want to monitor how does the microbial community change as a function of time, because that is the only way that the biochemistry of the system can be understood. Okay, so in panel C, for example, gives you distribution of the microbial communities. Now, I will just, in the next slide, I will explain to you as to how it is carried out. But the way you are going to interpret this data is that, for example, at 0 time, you have something, you have 60%. So relative, this is the terminal restriction fragment abundance. Okay, so now if one means 100% of the transcripts, which you have been able to see, out of which 66% or zero point 0.6 out of total 1 belongs to a community which is 562 lactobacillus. So all these colors which you see here in panel C are explained in this particular in this guide. So what you see here as pink
What is
color,
560?
it's a species of lactobacillus. Lactobacillus is a genus, but it's a very distinct species of lactobacillus, which has been named as 562. Okay, so it is Lactobacillus species 1. And what you see here in, for example, green, so we go back and we try to understand what is green here. So green is 516, and it is a Clostri DAC1, which is a very distinct kind of a microorganism. Okay, so while 68% at time 0, that is when the inoculum is actually, when the fermenter reaction is just starting at time 0. All right, you have nearly 66% of the microorganisms which are there. They belong to Lactobacillus species 1 and between like 0.63 to zero point 0.9. So it's about 28%. It belongs to Clostridiaceae. And then all these different colors, they define what kind of a microorganism is present, the relative abundance of that microbial system. So in other words, what you are trying to do is that not only are you trying to determine which microorganisms are present, but you also know how many of these are present at each stage. If I have 60% of these, then I have 28% of type 2, I have 5% of this, I have 2% of this, and I have 1% of this. So as you follow, the microbial community, you will see that the dynamics are changing. At this stage, at 21 days, when you are in the lactic acid, when you are in the L phase, for example, you have so many. It's really different compared to the distribution of microbes, which is at time 0, time 7, or time 14. So these very quickly, and I will explain to you as to how you really get this kind of a data. It's very easy to determine. You'll be surprised to see how easy it is to get this kind of a dynamics. Okay, so the first thing which emerges out here is that during the L phase, the proportion of the lactic acid community, because you see these symbols and the light pink, for example, here, 178 is lactobacillus, 562 is the maroon one, that is again lactobacillus. Okay, as expected, because you do have high lactic acid production, so it makes sense to say that during the L phase, the predominant genus which is there is that of Lactobacillus. All others are small. But then when you look at the gas phase, then you see the green ones, which are the Clostridia, okay, which are actually responsible for production of volatile fatty acids, because Clostridia are anaerobic microorganisms. And they break down sugars and they convert them into acetic acid, they convert them into propionic acid, etc. Because there is lack of oxygen, so they are not able to metabolize glucose or any sugar or any carbon source completely into carbon dioxide and water. So these are only partially degraded products of sugars, of other polymers, and these are the ones which are predominant in the gaseous phase. You see here, so how the population dynamics changes as a function of time and its correlation with either the lactic acid production or to the gas production. So is this clear to everybody?
I have a
Yes, ma'am.
question.
Okay, so very quickly now. So do you know what is this? What is this terminal
I have
restriction fragment polymorphism? How is it measured?
I have a question. Like I know that restricts RFLP is used for like human. So in that case, we measure like a satellite DNA, microsatellite, mini satellite DNA, etc. Microsatellite, mini satellite parts of DNA, etc. But in case of microorganisms, we don't see like that kind
Yeah,
of sequences which
yeah,
we find in eukaryotic cells. In
yeah.
microorganisms,
So
we don't have that kind of repetitive sequences. So why it is used?
okay, so let me very quickly go to the next slide. Now, is this okay, or should I switch to the larger slide mode? But if you see this, then I'm not going to come to the full slide mode. Can you see the slide clearly?
Yes, ma'am. Ma'am, I have
Okay.
one more question.
Yeah.
Ma'am, again, why we are using 16S rRNA sequences? We know that these sequences are almost conserved in across all species. So
No, no,
why
no.
are you using? Yeah.
Across, you see, across species, but not across genus. Across species, but not across. Let me, before I come to this slide, let me very quickly show you another slide before we talk about. Okay, so now, 16S, this is the gene which is coding for 16S. Then what is 16S ribosomal RNA? Okay, so it could be from a bacterium. Now within the gene, which is coding for the 16S ribosomal RNA, you see there are regions which are shown in dark blue, which are highly conserved regions.
Yes, ma'am.
All the ones which are highly conserved, and then there are regions which are shown in red, which are called as the hypervariable regions. Okay, so when you make a comparison between different genus, say Lactobacillus and Bacillus, these are two different, Lactobacillus is one genus and Bacillus is the other genus. Okay, you expect to see these regions are going to be conserved, but the gray regions are going to be the variable regions. Is that clear? And when you see species, so within Lactobacillus also, like you saw 562, 502, or you saw 162, the red is the hypervariable regions. Okay, so within species also there is going to be, so between Lactobacillus 152 and Lactobacillus 562, in the hypervariable region, you are going to see that which I have now sequenced, they are the same genus, but these are different species. So is that is that clear to everybody?
Ma'am, your voice was breaking. So can you explain the variable and hypervariable region? Your voice was breaking, ma'am.
Okay, I'm sorry. So let's show, quickly go over it again. So the what you see here in the blue is actually the gene. That is, it's a DNA fragment or a ribosomal RNA. The size of that resulting ribosomal RNA is going to be 16S. So that is, this S is the sedimentation coefficient. If you were to take that ribosomal, we can just say RNA, but the reason we say ribosomal RNA is because it is going to be eventually contributing to the formation of ribosome. It will not be translated into a protein. So that is a ribosomal RNA. Okay, so the entire gene, for example, you see is divided into these dark blue regions, which are called as the conserved regions. So if you are to compare the sequences of this particular DNA fragment between different genus, Lactobacillus, you have Bacillus, you have Clostridium, and you have, so many other Escherichia coli, you will see that blue regions are going to be conserved. But then there are the gray regions which are shown, and it shows variable region. So as you move from one genus to another, in the variable region, you are going to see differences. But then what you say as the hypervariable region, okay, is something among the species. They will be same in the blue region. So Lactobacillus species 1, Lactobacillus species 2 and 3, they could be similar here. They would be similar in gray, but they would be dissimilar in the hypervariable regions. So when you sequence and when you try to align, yeah. Yeah, please continue. I can't hear you. Or is it that my voice is not audible?
Am I audible?
Yeah, I can hear you.
I can hear you. Yeah. So, ma'am, my question is that, like, this on this slide, you have shown a fragment of DNA. Is this fragment is just coding for 16S rRNA, or this contains many different genes? I wanted to know.
No. It is just a single gene.
Ma'am, the thing is that. The thing is that in prokaryotes, we don't have RNA splicing. Then how can they manage to get protein out of this? In which like.
No. Okay. Let me, Ratnesh, we have to go back and we have to recall our fundamentals. Okay. So now let me show you this picture very quickly. Can you see the picture on the left here? It is a bacterial ribosome, which is 70S, and it is made up of two subunits, the 50S subunit and the 30S subunit. Okay, so you know that ribosomes, which are in the prokaryote, normally we say they are 70S based on their sedimentation value when they are placed in a gradient, sucrose gradient. The 50S is also called as the larger subunit, and the 30S is called as the smaller subunit. The 30S subunit is made up of 16S ribosomal RNA, the length of which is 1540 nucleotides, and it is made up of 21 proteins. So what you call as the smaller subunit of the ribosome is made up of two components. It's a nucleic acid and it also has proteins. So what you are trying to measure is the sequence of DNA. You are trying to sequence the DNA, which is going to code for the 16S ribosomal RNA. In in this smaller ribosomal subunit, both nucleic acid as well as proteins are together. So is that clear? Is that understood?
Yes.
Okay. Similarly, the larger subunit does also have proteins. It has 34 proteins and it also has nucleic acids, which are 120 nucleotides and 2900. So what we are trying to do is that we are trying to sequence that particular DNA fragment, which is going to code for 16S ribosomal RNA, the length of which is 1540 nucleotides. Okay. So in the picture, which I had just shown to you a while back, okay, so right up to here. So this is the first nucleotide of this DNA. And the numbers which you look at down below are 1243, 1435, and this would go all the way like up to 1570 or something. Okay, so this is the entire gene which codes. So within this gene, for example, there are regions which are called as the conserved regions, which are going to span from nucleotide 1 to 68, or from here to 136, or from this region to this region. So when you try to sequence this 16S, you can say coding DNA from different microorganisms, and when you try to align those sequences, there are going to be regions which are going to be highly conserved, but there are regions which are going to be variable or hypervariable. On the basis of that, you establish a relationship with three microorganisms based on, how closely they are to each other in terms of the variable and the hypervariable. So is that clear to everybody?
Yes, ma'am, it is clear. Actually, this question was like in our one of the molecular biology examination that can we get differentiate between us two species using 16S rRNA sequencing. So
Absolutely. See, because if they belong to the same genus, I think the cutoff they have put is 97%. Okay, so if the two sequences are similar by 97%, you will say it belongs to the same genus. But if the similarity percent, 93%, then 90 different genus. That is the rule of thumb, which is for phylogenetic classification.
yes, ma'am.
All right, so can we move forward now? Can we go forward now?
Yes, ma'am. I have one more doubt. Like we know that there is last common ancestor, last universal common ancestor. So we know that I know that like in bacteria and in eukaryotes and in archaea there is 42 like around 40% genome is similar in all across uh, all the domains. So which kinds of genome are that?
Well, I have to read back. I have not read on that subject for a long time. So let me read and I don't know, they are some of the most primitive, and maybe Microbacterium is one of them, or Haemophilus influenza, maybe, of course, that's a virus, that's a viral genome. But I will have to read up and come back to you to see, to see how they have digressed with respect to time. So now let's go back and So in order to give you an idea of how such microbial dynamics are monitored, for example, through what is called as TRFLP approach to community fingerprinting of the genes. Okay, so this is actually the terminal restriction fragment length polymorphism. It can also be applied to bacterial identification, bacterial classification. So what you do is that once that sample is extracted from phase 1, on different days you have extracted a sample and so from the solid pellet, from the cells, you centrifuge out that material, you collect the cells, they were stored at minus 70 or whatever. You can extract the DNA from here. Okay, so the soil can be suspended or this solid digestate can be suspended in phenol, proteins can be gotten rid of, nucleic acids can be precipitated by adding ethanol. So you can see very nice and beautiful DNA fibers which are precipitating out. You must have done this experiment in the second year also, that you can extract the DNA. So the DNA is extracted from the community. It's taken in a small test tube, Eppendorf tube. You carry out PCR with the fluorescently labeled 18S ribosomal RNA forward primer. Okay, so what you do now, so what you have inside, for example, is now you have extracted the DNA. The DNA is going to be denatured, and now you have a primer which is fluorescently labeled. Only one of the primers is fluorescently labeled, which is the forward primer. Okay, so you see now what you see here, for example. So 1, 2, and 3, these are 3 different DNA molecules, and in the first cycle of extension, you have a fluorescently labeled forward primer. Okay, that means the 5' end of that particular primer is fluorescently labeled, and you have an extension which is going on. So it's going to bind at this particular location, and you have an extension reaction. So in all the 3 DNA, let's say that these are 3 DNA molecules. So with the forward primer, this fluorescently labeled primer is now going to anneal to one of the 2 strands of DNA, and it will be extended in the presence of DNTP and the Taq polymerase. Now, one of the 2 strands of the DNA is now going to be labeled with. Okay, the next thing what you do is that you carry out restriction digestion of the PCR product. Okay, now consider that these 3 molecules, they belong to 3 different bacteria. Okay, so when you carry out so in this particular case, all the DNA fragments which they have obtained, okay, they are fluorescently labeled, number 1, and they are subjected to digestion by one enzyme. I think it was MSP1 or something. Now, because they originate from 3 different bacteria, the chances are that this MSP1 site is going to be at different locations in these 3 DNA molecules which are amplified. In the first case, let's say the MSP site is here, so the DNA molecule which is amplified is going to cut here. In the second bacterial DNA which you have amplified, the MSP site could be here. In the third bacterium, it could be located here. Okay, so therefore, the fluorescently labeled DNA molecules are going to be different in length. So in this particular case, the length of the fluorescent DNA, which is now labeled as A, is going to be much longer. The one which you see here is going to be shorter, and the one which you are going to be seeing here is going to be very, very different. Now, after you do the restriction digestion of this DNA, all the products are going to be run on an agarose gel. Okay, so the DNA molecules are separated completely, and the ones which are fluorescently labeled, they can be visualized. So you can just irradiate it with the light of a certain wavelength, and the ones which are fluorescent DNA bands, which are going to be for this band, this or this band, the fluorescently labeled will become immediately clear to you, whereas the other part, because they are not fluorescently labeled, they are going to be in the gel, which can be visualized by ethidium bromide, but they will not be fluorescent. So this heterogeneity in the length of the fluorescently labeled bands, okay, will give you an idea of how many different types of bacteria are present. So in this case, it is an indication that when you started in the community, there were 3 different types of bacteria here. So if you get 3 different fluorescently labeled bands here, they are indicative of 3 different members. in this community. At the same time, now you can also you can read each one of the bands through a fluorimeter, where the intensity of the fluorescence can be measured. Okay, so in the you see here is the relative fluorescence and recognition of the labeled fragment. So when you pass a fluorimeter over here, now the more the members of microbe A are, the more you expect, because they will also have more DNA to contribute. So the fluorescence that they will contribute will be much higher compared to that which is contributed by B or C. Okay, so you expect to see a profile like this. So the relative fluorescence of C is going to be much more here. So this one, if the fluorescence of this is much more here, then you also have a way of quantitating it. That
Ma'am,
50, yeah.
ma'am, the relative fluorescence is the intensity or is it the visibility, that if the more number of DNA of C are there, that means more
Yeah,
visible it is,
exactly.
and that's why.
Not
Okay, ma'am.
visible, but in a fluorescence, you measure the fluorescence units. The meter is going to show you 5,000 units, 7,000 units. In a fluorimeter, you can actually measure the fluorescence. Okay, so
Yes,
depending
ma'am.
on if you read 10,000, that means the concentration of this particular bacteria from which this band is emanating is much more compared to this. Okay, so this relative fluorescence, therefore, so everything you just add up, like, you say relative fluorescence, here you had maybe 20,000 units, and here you had kind of 7,000 units, and in A you had kind of 10,000. So you add them up, 20 plus 10 plus 7, 37,000 units. Then you say 20,000 divided by 37,000. So so out of 100%, how much is being contributed by C? And that is exactly the data which you see in this particular picture. When you see on the y-axis in panel C, the relative terminal restriction fragment abundance. So out of 100% of relative fluorescence, this particular signal, okay, is contributed to by Lactobacillus 562. Okay, so is that clear as to how both identification can be done and enumeration of these can also be done? That is, they can be quantitated.
Can we full screen?
Yeah, I'll go to full screen. Yeah, I'll go to full screen. And if there is an issue, let me know if you're not able to see. Okay, so can you see this now?
Yes, ma'am.
Okay, so
Yes, ma'am.
relative terminal restriction or relative fluorescence, terminal relative fluorescence abundance. So whatever signals which you saw, in that slide which I projected, they will all be added to make up one. And then each one of them can be now quantitated: 20,000 divided by 37,000, 7,000 divided by that, 10,000 divided by 30,000. So this is the way each one of these can be now enumerated, and their distribution can be mapped over a period of time. So you can do a community profiling. So this I've already explained to you, and I hope you understand. So, make sure that, it's only one of the two primers. One of the two primers, because for a PCR reaction, remember that two primers are required. Okay, one is called as a forward primer, other is called as a reverse primer. But here only the forward primer is going to be fluorescently labeled. So you will only have, the ones which are extended with this is going to be labeled. But the other one, the reverse primer, it's not fluorescently labeled. So those amplified products will not be visible at all to you. Or they will be carried out, but then since they don't have a fluorescence, they will not be picked up, but it doesn't matter because those DNA molecules anyway are going to be picked up by them. Okay. So again, just to give you an overview, that the bacterial community dynamics was also investigated in the second phase of the reactor. That is, which is the one where you have the acetogenesis and you have methanogenesis. So again, to give you an idea of how this time profiling can be done. And now what is very, very interesting is that You can also make a correlation analysis of the methanogenic community with the process data of the second phase reactor. So many things are here. You have all the raw data in front of you. So what you can try to do, for example, is that these are methanogenic terminal relative fluorescence. Okay. So the chart which you see down here, for example, so the color codes which are defined here is the correlation data. Okay, so if you see a color like this, the maroon one, okay, so the maroon one refers to a negative correlation, -1. But if you see a +1, that means a positive correlation. Okay, so the light green one, for example, so this matches somewhere here, so the correlation is 0.4. But then if we look at this particular one here, okay, it matches with this color, so the correlation is 1. But the correlation between what? So the correlation is now between a process parameter and the presence of a given microorganism. So let's take the example here. Okay, so there is a correlation. So these are the abiotic parameters of the second phase bioreactor. So if we were to monitor the pH value, which is existing at that particular time, okay, with, for example, with the presence of this 163 species, 163 microorganism, there is a very good correlation here. Okay, I hope you're able to understand the data. It's very valuable. So each one of...
Ma'am, just one doubt. Ma'am, the green one is in the range of 0.8 to 1. So, ma'am, what exactly it is depicting, ma'am?
Okay.
The pH value in reference to pH
No.
value.
It means the correlation between pH value and the presence of 163 is 0.8. So every time the pH value goes up, there is more of 163. That is a positive correlation. But if you look at acetic acid, it comes in the pink, where it comes in this color, light maroon, where the correlation is actually -0.6. So that means every time the acetic acid concentration goes up, the presence of 488 microorganism goes down. So there is a negative correlation between acetic acid and between the presence of 488 micro.
Okay, ma'am. Okay.
Is that clear?
Yes, ma'am.
Right. So now, because, now you have all the data, you have the process parameters, you have the performance of the reactor, and you have all this data, you have a microbial community data. Certainly you want to make a correlation because eventually your target is that, yeah.
Ma'am, it implies that the if more of acetic acid is formed, that means the 488 organism will decrease or vice versa. Is it that we can interpret?
Absolutely. Absolutely. Now, if it is an anaerobic digestion process where you're going to monitor methane, you want to keep your eyes on methane, basically. You're carrying out, you have this maize silage, this material, which is the waste material, and basically you want to use it for energy production. Depending on what is the value of poultry, I think because animal poultry is a very big industry. So normally we take all these agricultural byproducts like rice straw, wheat straw, and we feed it to the cattle because we have a huge poultry business. Because some countries in the world may think, well, energy is more important to me, I want to make methane out of it. You want to keep your eyes on the methane. So now let's look at, for example, where is the correlation? So between the presence of methanocoelius, this particular microorganism, which is actually a methanogenic bacterium, all right, and the methane yield, there is a huge correlation. Okay, so in other words, what it indicates that in the bioreactor, at that particular time when the data was collected, you must have more of methanocoelius, only then your methane yield is going to be more. Okay, similarly, for example, methanosarcina, which is another methanogenic genus, the correlation is 0.2. So, the pH value, for example, there is all right, the bacteria seems to be growing, methanosarcina seems to be growing well. If I try to correlate it with the with the conversion or any other process parameter here which they should have shown. But in this case, methanosarcina, for example. Okay, so how it is correlated, the presence of this, how is it correlated to different process parameters? Okay, because eventually what you are going to monitor in a bioreactor are, it's very expensive to do a microbial community profiling, but it's not expensive to measure methane in the outlet. It's not expensive to do carbon dioxide, gas, the pH can be monitored, or the hydraulic retention time is something this thing, organic loading data, etc. So many of these process parameters are now being linked to the presence of unique microorganisms in a good methanogenic bacteria, so that your objective as an engineer is to be able to monitor these process parameters, which will ensure that only certain methanogenic bacteria are going to survive, which have been found to be correlated to methane production in the bioreactor. Okay, so I'm going to monitor, and I'm also going to make it every time a reproducible methane producing bioreactor. Okay, so is that clear to you?
And ma'am, one thing I have to ask, ma'am, that the dark green one, it shows that
Yeah.
more the rate as compared to light green, more
Yes.
production. Yes, ma'am.
Definitely. You see, because you just have to compare the color codes on this particular scale, and each one of these squares only refers to the correlation data. But how do you interpret it? Okay, so if you see green, that means there is a positive correlation. But correlation between what? Correlation between the pH value and the presence of 163 microbe. Okay, so the conversion into what? Into maybe methane production is very high when 163 is present. So here is these are the process parameters which you are measured, or the metabolites which you have measured, and here is their correlation to the different microbes which you have now been able to enumerate in the second phase reactor. So is that clear?
Yes, ma'am, it is clear.
Okay. So now, again, very interesting in this table from the same paper. So again, we have the correlation of abiotic. So they have kind of summarized it now. That is the correlation of the abiotic parameters like, the pH, or production of fatty acids, or the hydraulic retention time, organic loading rate in the first or in the second phase reactor, with the bacterial terminal relative fluorescence correlating positively with all these methanogenic bacteria. The red color indicates a negative correlation and the green color indicates a positive correlation. In other words, if we look at the presence of methanobacterium now, okay, so this particular microorganism, which is actually responsible for methane production, is negatively correlated to presence of propionic acid, valeric acid, hydraulic retention time, degree of conversion, and pH, but it is positively correlated to presence of butyric acid. So in other words, in the second phase, if the concentration of butyric acid is more, then the chances are that I will get more of the methanobacterium and my methane yield is going to be more. Okay, correlating the abiotic parameter of the percolate,
Ma'am,
which is
the
actually in the first phase. Yeah. Yeah,
what
continue,
is
continue your
the
question.
color depicting? Ma'am,
Okay,
what is the color? Okay, I understood.
understood. Okay, so green is a positive correlation, red is a negative correlation, and NC means there is no correlation between any abiotic parameter and what happens to the presence of methanobacterium. There is no correlation here. Okay, so overall, it gives you a very good idea that finally methanobacterium, that is methanocoelius, is positively correlated to methane production and degree of conversion, and conversion is related to CO2. So if there is too much of a buildup of carbon dioxide, then chances are that the concentration of these microorganisms will fall low, and eventually the methane yield is going to be compromised. So the idea is to be able to remove at this stage, do we have a process for stripping carbon dioxide from the reactor at this stage? Do I have a process for stripping carbon dioxide? Because that is going to negatively affect my methane producing microorganisms. So this is what I learned from this data as a process engineer, and this is the way I'm going to operate my reactor.
Ma'am, based on the data, we can manipulate things like CO2 can be, we can efficiently reduce CO2 so to grow the methanobacterium of
Absolutely.
to grow the. Yes,
Absolutely.
ma'am, understood.
Or like acids, for example, if there is a building of too much of acid, you may want to control your feed rate. That if you were supplying your feed at 5 gram per hour or so many liters of that slurry, you may want to slow down on the feed rates, because all these acids are going to inhibit my process. It is positively correlated to organic loading rate. I may want to give, this could be slow, my feed rate, but it could be more frequently done. Organic loading rate, if it keeps on increasing, that provides you with some kind of a guideline. But this is always hit and trial every time, unless we really learn enough about the system to be able to achieve the reproducibility. Okay.
Ma'am, in the previous slide,
Yeah.
ma'am, ma'am, it was the theoretical data or the experimental data from
No,
where we are interpreting. Ma'am, yes, ma'am.
this is all experimental data, which is from the same paper where they have set up these two-phase bioreactors. I don't think the results are contradicting each other, that what is being shown here is contradicting, except that in this particular slide, okay, the focus is only on methanogenic bacteria. There are only 3 different species of because eventually your objective is to be able to increase the methane yield. So the data is kind of extracted from the previous slide, except that you're only looking at now these 3 different genus and trying to correlate it. More broader picture, that data was incomplete. There was no relationship given to volatile fatty acids, etc., which is brought in here. Okay, so is this clear to everybody? Should we move to the next slide
Yes,
now?
ma'am. Yes.
Please read that paper, because we talk about so many, we have talked about subjects like microbial ecology, microbial interactions, so on and so forth. But when you look at this particular case study, you see how everything has to be put in the proper perspective. Why do you need to know about microbial ecology? Well, because in a process like wastewater treatment, in a process like soil remediation, there are going to be community changing as a function of time. So you need to understand what kind of biochemistries are they doing. Are they helping each other? Can I make a synthetic inoculum which can be supplied? I can be manufacturer of an excellent inoculum, supply to people who are setting up a wastewater treatment plant, because I know already. And you can also supply the guidelines that, well, this is the way the reactor should be operated at crude level, so on and so forth. Some principles which we have discussed in this paper, but which I just want to put in so that everything is brought back into your mind. So we saw that the changes in the microbial communities are occurring, as you saw, very, very dynamic system. These are both biotic, which are the living, as well as abiotic factors. They drive the evolution of these communities. So their composition is going to change, and these are very dynamic. There is a natural succession. There could be a climate change, or because of human activities. Plant or in an activated sludge process, there is a natural succession of these. Okay, so you're not doing anything in the activated sludge process. The reactor has been running. You're only recycling back 30% of the sludge back into the reactor. Okay, so these are kind of natural succession. What abiotic factors affect redox, temperature, the biotic pressures are imposed by different populations which are competing for either the same organic carbon or nitrogen. Okay, so there is a competition among the microorganisms also because they are fighting for the same resource like carbon or nitrogen. So soil contains about 10 to the 8 or 10 to the 10 bacteria per gram based on direct count. Okay, so if you were to take 1 gram of soil and you find out how many bacteria does it contain, you can do a dilution, etc. And you can simply either look at it under the microscope and you can make a 100-fold dilution, make a 200-fold dilution and just look at it under the slide. So you will be able to see how many bacteria are going to be present here. If every population is present at 10 to the 6 per gram, this would mean that there are 100 to 10,000 different populations per gram of soil. Okay, so mind you,
Oh,
there
ma'am,
are 100
this
to
data
10,000.
like 10 to power
Yeah,
10 to power 8 to 10 to power 10, is it for everywhere? Like I can if I collect data from here and from somewhere else, so that will be like in this range, or this
absolutely,
is or
absolutely.
is okay, ma'am.
Hmm. Generally, it is. And you'll be very fascinating to know that the more fertile the soil is, the better the soil is, the more the diversity is going to be. Okay, so if you look at land patches where agricultural practices have been going on and people have been putting fertilizers and what have you, there is too much of nitrogen, too much of phosphorus, the soil has become alkaline. If you look at the microbial community composition, the diversity has gone low. Why? Because the diversity has gone low. Serves index of health of the soil. Okay, so I will come to that. I have a picture to show you. So I'll show you this data which is taken, the soil the diversity. So anyway, so these populations, they exist next to each other and therefore they are forced to compete or work synergistically for resources. So how do the microbial communities respond to the biotic pressures? Very, very interesting. So either they acquire genes from the environment which allow for previously unavailable activities. Okay, so when you say acquire genes from the environment, okay, now many microorganisms, when they like Pseudomonas, for example, Pseudomonas is present in soil, for example, and there are some plasmids and things like that. So when these microorganisms, they lyse, for example, they also contain plasmid that is excreted into the soil, because when the bacterial cell lyses, the plasmid DNA is also released into the soil because of process of conjugation or transformation or whatever. These genes can laterally transfer from one microorganism. Okay, so a bacterium like Pseudomonas, which was not able to grow on a soil which was contaminated by a hydrocarbon, is capable of growing now. Why? Because it has a plasmid from another microorganism which was capable of degrading a hydrocarbon. Okay, that's what we mean. That microorganisms in response to such biotic pressure, because let's say you have a soil which is contaminated with hydrocarbon. So now this poor Pseudomonas doesn't have any gene for metabolizing this hydrocarbon. Okay, but in the same vicinity, okay, there is another bacterium which makes this thing which can metabolize hydrocarbon, but that particular gene is present on a plasmid. Then the Pseudomonas can acquire the ability to metabolize that. Okay, so how have they responded to this biotic pressure? That is, when they are competing for the resource, they can acquire genes from the environment, which allow for previously unavailable biochemical activities. Okay, so for example, so how do the genes come? Either point mutation events which alter the regulation or kinetics of enzymes. So within this Pseudomonas, there could be a gene like naphthol, okay, except that it wasn't working too well, but then okay, if there is a mutation in that gene, that particular microorganism which was capable of mutation.
Ma'am, your voice was continuously breaking. So Ma'am,
It,
please can you hear
Yes,
us?
ma'am. In last 2 minutes, it is breaking.
Yes, ma'am.
okay. So now, all right, let me come back again. So is it better? Can I be heard? Can I be heard better?
Yes, ma'am. Yes. Yes, ma'am.
Okay. Okay. So now I was giving you an example of Pseudomonas. So now the Pseudomonas, it finds itself, it's a microorganism, finds itself in a soil, okay, which is contaminated with naphthol, which is a hydrocarbon. Okay. Now in the same atmosphere, in the same soil, there is another microorganism which contains a plasmid, which codes for gene which can break down naphthol. So this microorganism dies after some time. The plasmids are released into the soil, and that plasmid can be taken up by Pseudomonas through transformation. Okay, there are so many events which allow this genetic exchange to occur. Okay, so this enters into Pseudomonas, this. So acquiring that plasmid, for example, is going to confer the ability to degrade naphthol in the Pseudomonas. That's what we mean by when we say that in response to such biotic pressures, there is a competition. There are different kind of microbes, and if the Pseudomonas doesn't know how to metabolize naphthol, it is going to be eradicated from that community. Okay, so how does it fight back? It acquires the ability to hydrolyze, to break down naphthol by virtue of having a gene, which it acquires, through transformation, or if we say through lateral or horizontal gene transfer. Horizontal, what we mean is that there is a conjugation event which is going on. The DNA can directly enter through transformation event, so on and so forth. Okay, or there is an existing gene in the in this Pseudomonas, but the activity is very, very low. It is not able to metabolize naphthol, very, very slow process. However, there is a mutation in that particular gene, as a result of which it can alter either the regulation or the kinetics of that particular enzyme. Okay, so that enzyme, maybe its kinetic is changed or its Km is now decreased. So it is able to bind to that substrate far more effectively. And as a result of that binding, the rates of reactions are going to be increased. So now this Pseudomonas is able to compete in that environment. It is able to metabolize naphthol. So it can compete with other kind of microorganisms which are able to break down. Therefore, it can survive. So is that clear?
Yes, ma'am.
Okay.
Some
So
plasmids
there are
that
two
have
scenarios
entered
which are
into
given.
the Pseudomonas.
Yeah,
Ma'am,
through
the plasmid that
transformation.
entered.
Yeah, sure. Complete your sentence.
Yes, ma'am. So, ma'am, I was asking that the plasmid that was entered through transformation in Pseudomonas that will degrade the naphthalene and that will completely eradicate
Absolutely.
naphthalene.
Absolutely. It makes extracellular enzyme because the gene which is present on that plasmid synthesizes an enzyme which is extracellular. It gets out of the Pseudomonas and it is able to metabolize it to a stage where other enzymes from Pseudomonas are able to carry out the rest of the function. Okay, because many of these, as we are going to learn, as we are going to proceed through the course, you will see that many of these hydrocarbon degrading enzymes, interestingly, they are coded for by plasmids. Okay, and they're coded for by plasmids.
Yes,
And
ma'am. Understood.
similarly, antibiotic resistance genes, many of these are coded for, they're, the genes encoding them, so to speak, are present on plasmids, not on the chromosomal DNA of E. coli. And that is the reason why there is so much of trouble. And people talk about multiple drug resistance because, you have all these plasmids, you have all these streptococci or whatever, which are antibiotic resistance now, because all these genes have entered from other microorganisms. So in the hospital scenario, the common infection is streptococcus, the common infection is E. coli. Okay, but there are many these drug resistant genes which have now entered into these hospital bacteria. And today the fatality because of hospital infection is so high because streptococcus is no longer responding to that antibiotic. It has become resistant. And many people, unfortunately, they die of acquiring these infections.
Yes, ma'am. Ma'am, actually, I was in one paper like that I got in news, so article. So in that paper, that was about 2014. Something kind of E. coli was being some different strand of E. coli was came out in Germany, and that was killing very high number of people. Even
Absolutely.
like yeah, even higher
Absolutely.
rate, even higher than cancer, etc. So. In that paper, in that context, I had a article like there was a video, and in that they were explaining that we are not able to like how to determine this species and something like that. uh, they can mutate very fastly or something.
Mhm.
So
Yeah, yeah. But by and large, today, if you look at any standard microbiology book, okay, they will keep cutoff at 97%. But sometimes, there could be two different species which could share you have to make a cutoff. For bacteria, you keep it at 97%. Now earlier, when you were doing eukaryotes, comparison, you were doing the 18S ribosomal RNA, because eukaryotes make larger ribosomes, and their smaller subunit is also the 18S, not 16S. Now earlier, the 18S ribosomal RNA sequencing was done in order to make a relation between the different eukaryotic systems. But today they are also doing other sequencing, what is called as this IRS sequences, which are in between these ribosomal genes, the intra ribosomal genes, smaller segments. So other parts of this gene clusters are also being sequenced in order to establish a relationship between fungi. But in bacteria, we don't have a choice right now, because there are no introns there. The genes are completely intron free. And the only way you can sequence 16S is the most reliable method for sequencing them. There is no other method which is available.
yes, ma'am.
Morphology and other things is not really, but at nucleic acid level, I think this is the only thing which is reliable right now for bacteria. All right. So now, anyway, let's get back very quickly to so responding to such biotic pressure could be carried out by acquiring genes from the environment, as I explained here. So a large number of population and their rapid ability to respond to environmental pressures, therefore, results in an ever-changing and ever-evolving microbial community within any ecosystem. Okay, because conditions are changing, the dynamics are changing, the nutrient inflows are changing. So as a result of that, the microbial community is changing. As I said, in healthy ecosystem, the diversity is very high. In stressed environments, extreme environments, which are very specific selective pressures like the pH or very high temperature, the microbial diversity is very low. Then the question becomes, can diversity serve as an index of monitoring the health of an ecosystem? Now, uh, so microbial diversity, again, just the way the sequencing has been used in order to enumerate, it can also be used for understanding microbial diversity. Okay, so it is kind of overlapping with what I told you earlier, except that in that paper, what we were looking at was the restriction fragment length polymorphism. But then if you were to sequence, after you amplify this 16S ribosomal RNA, and if you sequence them, then you do exactly, you are able to figure out which genus it is, which species it is. So it allows you to make a point on microbial diversity. Is that clear?
Yes, ma'am.
Okay, so let's quickly move. So this you have already understood, and my idea of showing this again here is that whenever you do this, you, for example, isolate your DNA from a sample, then there are companies which are providing these primers. Like for example, you have a primer like 27F and 338R. Okay, so this is the forward primer, this is the reverse primer, and it is the primers are designed in such a manner to be able to amplify the V1 and the V2 region. Okay, and what is the length of this amplicon going to be? It is going to be 312 base pair. And it can also be used for microsequencing, and it is supplied by the company Roche. Okay, so this is like Roche, on Roche 554, sequencing can also be done if you use this forward primer. So, you know, it's like it already has a number. So if I have a sample, I don't have to design my own primers. I can simply order these primers because they are commercially available. Okay, so you can use this 27F338R in order to sequence this region, amplify, sequence it out on a Roche sequencer, and on the basis of that, you will be able to assign, identify it, do a sequence comparison of the DNA fragment and say, well, it belongs to this genus and it belongs to this species. Okay, so all of these are commercially available. Okay, so in that in the paper which I discussed with you, they have used this 341F. So there is a forward primer which is binding at 341 nucleotide, and there was a reverse primer which was binding at this particular location. Okay, so what you see here in red, for example, are the primers which were selected, which were designed by those group of people, and they had amplified this particular sequence in order to identify that microorganism. So apart from looking at the distribution of different microbes, you are also able to identify them only by virtue of sequencing that fragment which you have amplified. Now, in addition to this 16S ribosomal RNA sequencing, many people are also doing what is called as multilocus sequence typing, where there are other genes which are also going to be taken up and they are going to be sequenced. Okay, so imagine that this is a bacterium and there are ones which are marked in red. This is locus 1. By locus, what we mean is that this is where one gene resides at this particular location on the bacterial chromosome, which is shown as a circular DNA molecule. Okay, so right here, 1, 2, 3, 4, 5, 6, and 7. Okay, so if you have primers which are going to be specific to these DNA sequences, you are going to amplify these 7 DNA fragments. You are going to sequence them and assigning an allele via the World Wide Web. Okay, so now once you have sequenced this particular locus, let's say it is the gene for tryptophan A part of it, because I find that among the different bacteria, this is something which shows considerable variability. Okay, so I'm going to identify it as a target, sequence it, and then I'm going to see how much it, where does it really belong to? What is the identity of this? Okay, so it is assigned a certain number, which is called as 4. Then the second DNA fragment which I have identified is here, and I sequence it and I find out that, well, it can be assigned this particular allele number because there are only so many variations which are present. So I assign it to allele 4 or variation number 4. Okay, so for all these 7 loci, there is a code which I'm going to assign. So this is like my variant 4, variant 4, variant 2, variant 1, and 1. On the basis of that, this particular microorganism will also have, so ST means it is called as the sequence type. So if it shows 4 variant here, 4 at the second locus, 2 at the third locus, 4th at this locus, then this variant will be called as the sequence type 81. So I'm able to establish, apart from 16S ribosomal RNA, I'm also able to identify it as a unique microorganism. So there are 2 ways of classifying now, both 16S as well as the sequence typing. So is this clear as to what is being said here?
Yes, ma'am.
Okay. So now, I just brought to you the subject of microbial diversity. Okay, so very quickly, how do we measure microbial diversity? Is it something which can be quantitated? And the answer is yes. In order to define microbial diversity, let me just okay, so there are different parameters, which have been numbered, which are called as are called as, for example, alpha diversity. So you have a population, you have a microbial population which contains different types of microbes. Okay, and alpha diversity represents the species diversity in a sample unit, and it is determined using species richness or a diversity index. Okay, each one of these terms has a special meaning, and I'll come back again to explain that. The diversity can be either alpha diversity, you can say, or you can measure it in terms of the beta diversity, gamma diversity, species richness, etc. Okay, now let me come to this particular figure and explain to you that diversity is a very, very, I think, there is no other term for it than what can be understood in ordinary English. Okay, diversity means that in a population, how many diverse kind of microbes are really present. As simple as that. Okay, so alpha diversity, for example, um, so So here is the site A. It's a microbial site A. Okay, so you see there are three different types of microbes which are present. One which is shown in black, the other which is in orange, and the other which is in green. So alpha diversity will be 3 here. As simple as that. Okay, however, when you say species richness, that means how many of each type are present. So species 1 is present at 3 number, species 2 is present at 2 number, and species 3 is present at 6 number. So when it comes to species richness, you say that this particular species is the one which is most represented in that particular sample. Okay, similarly, the site C. Okay, alpha diversity is only 2 here. Species richness, yes, this particular species is more abundant compared to the other one. So that's what we mean here. So when you say alpha diversity, it means the species diversity in a sample unit, and it can be determined using species index or a diversity index such as alpha. Now the beta diversity is called as the difference in the diversity. So if the diversity in sample 1 is 3, if the diversity in the sample is 2, then what is the difference in the diversity between the two? Okay, so now going to this slide, for example, so here I have a little bit of a confusion I will come back to later on. So in this particular species, the diversity is 3, and here the diversity is 3. So the beta diversity between A and B, because here what they have done is that they have taken a difference 3, okay, and I still have to figure out why they have taken 2 here, because the difference in the diversity should have been actually, I don't know why they are using the number 2 here, or it is the minimum representative of a species. I have to come back and, I will clear it in the next class for you. I don't know why, because, 3, and here also the alpha diversity is 3. So, but they are still saying 3 minus 2 plus 3 minus 2. Or is it how many of that particular species are present? So if there are 3 which are present here, 3 minus 2 and 3 plus in each case. So anyway, I will come back to this point in order to. So are you still around? Are the students listening?
Yes, ma'am.
Yes. Okay, so there is a little bit of a confusion in the slide. Maybe I will choose the right slide in the next lecture. So in this figure, let me quickly go back and refer to this. One of you had a question that if we look at, for example, the diversity, okay, on the x-axis, what you see is the 16S ribosomal RNA richness. Okay, so in other words, by richness, what we also mean is how many of these species have you been able to identify? Okay, so you have, for example, either 4,000 or you have 16,000. So, it is not this, it is not how many of each kind are present. It is only how many, it is like how many genus are present. Okay, not how abundantly one species is present. That is what 16S ribosomal RNA richness means. Okay, so if the number is 4,000, that means in a population, okay, there are 16S ribosomal RNA sequences, which are 4,000 of these are present, which are different from each other. And that is what the number 4,000 here indicates. In a population, if you are able to read 6,000 different types of 16S ribosomal RNA, so that is what the number here is going to represent. So is that clear to everybody?
Ma'am,
The
can you explain
triangle
again?
and the shapes, what exactly they are
Oh,
depicting? That
yeah.
the
So
maroon
I
color shape, that is
yeah,
around 2000 in number.
right. I'll just come back to that. First, let me finish the information on the x-axis. On the y-axis is called as a metagenomic richness. Okay, that means that if you sequence these genes, see what you have done on the right. In order to get the data for the x-axis, you have only sequenced the gene for the ribosomal RNA. You don't know anything about that microbe. But then in the metagenomic richness, what you have done, you do a functional analysis. You do the sequencing of the entire fragment, which is indicative of what kind of functions, what kind of biochemistry does it really contain, this particular DNA fragment. Because anytime when you clone a gene from such a microbial population, you have a microbe, and then you are doing, when you make a metagenomic library of that, you are practically sequencing the entire genome of that microorganism. So this represents the functional diversity. And this represents only the diversity, which is at the 16S level. So it is a single gene, and these are the multiple genes. This is what the data on the y and the x-axis refer to. Okay. And what do we have here, which are the inverted triangles? This refers to the microbial communities which have been sampled from cold desert. So if you take from cold desert your soil samples, you find that it is not so diverse. Only 5,000 different type of microbes are present there. However, the functional diversity is quite high. 2,200 different type of functions are being performed. So more of similar type of microorganisms. So you can say that the microbial diversity here is much lower, but species richness is more in the cold desert. In the blue sample, this is the sample which has been connected, soil sample, connected from Arctic tundra region. Or the green are something which are tropical forests, or the bottle green are the prairie which are in the South America, which are the tall grasses, the savanna grasses, etc. And red is like a hot desert. Okay, so somebody has gone on to do all of these to see that what kind of diversity exists in these different habitats and what kind of functions exist in these different habitats. And generally, as you see, that the correlation is really very low. It's not that as the species richness goes up, you are going to get functionally more and more genus, more and more functions associated with that. The answer is no. And that is something which is shown to you here. So the relationship between functional redundancy and and of course, and what does it have to do with ecosystem stability? So is this clear to everybody? Are people following what is being said here?
Yes, ma'am, but like I could not get what does the richness mean here.
Uh, you see, by richness, what we mean is that, let us say, in a microbial community, there are 5 different microbes which are present, but one of them is 1,000-fold more abundant compared to the remaining 4. That is what is species richness. I hope that
Yes,
should
ma'am,
be clear
understood.
to you.
Yes,
Okay.
ma'am, understood,
All right.
understood.
So now we are trying to find out, and of course there is the species richness, there is also the functional diversity, and now we are trying to correlate these parameters to the ecosystem stability. In other words, does diversity have anything to do with stability of an ecosystem? Again, it's the fundamental question which we were trying to address. Okay, so this particular picture will show you the answer to that. So on the x-axis, we have a species richness. On the y-axis, we have ecosystem function and ecosystem stability. Okay, as species richness increases, as you see here, okay, look at the first curve, which indicates the functions. Okay, obviously, as the species richness increases, you can expect that the functions that this community is able to perform are also going to increase, because you're going to add new kind of genes, new kind of functions, so on and so forth. However, beyond a certain point, when you increase the species richness, okay, the functions are no longer going to be increased, because basically you are adding more and more of the same microbes which are able to perform exactly similar function. Okay, you may be creating diversity at taxonomic level, but you are not creating diversity at the functional level. Okay, but does it have anything to do with the stability? Then you see, as the species richness increasing, the stability also starts to increase. Okay, so therefore, what it means is that, you know, as species increase, as microbial diversity increases, the functions may not increase, but the stability of the ecosystem increases. So the more diverse an ecosystem is going to be, the more stable it is going to be. And that makes sense, because if there is a natural calamity, and there were microbes, there were 10 microbes, out of which 4 were very sensitive to pH. Now, if there is a sudden pH change in that microbial community, you get nutrient and some kind of effluent comes in, which is highly alkaline. So these 4 microbes are going to be wiped out because they were very pH sensitive. But there were 6 other microbes which were able to carry out the same functions. They were not so pH sensitive, so that function will still go on and the community is going to survive as a whole. So in other words, the microbial diversity may not add to functional diversity, but it certainly adds to the stability of the ecosystem. So in other words, microbial diversity can serve as an index to determining stability of that ecosystem. And that's the reason why. Is that clear?
Stability will be get saturated after some point of time at this richness of the species.
Sure. Like you see here, that the stability also is plateauing off. It's not really increasing the way the increase is shown here, but after some time. But yes, compared to functions, functions have particularly flattened out. Okay, but then you see that the stability, because now, it's very interesting. Like when you say, today, of course, we have become so aware of monitoring our rivers. So the Ganga project, the Yamuna projects are very big. So what they are also trying to do is that as a source of indicator of the health of Ganga, they collect water from Gangotri. Okay, they collect then somewhere along the line. They only determine what is the microbial diversity. So as Ganga moves from Gangotri to the rivers and to the other areas, okay, the microbial diversity index of those samples can serve to indicate, they serve as indices of health of Ganga. Because as we said, that microbial diversity is indicative of a very healthy ecosystem.
I'm sorry to interrupt, but our next class has already started.
Oh, please leave. And I'm also going to stop now.
Yes, ma'am.
Yeah. Okay. So I'll see you next week, and I'll upload these presentations.
Ma'am, just one short question I have. Ma'am, how does it how does it get stabilize the Ganga? I don't understand, ma'am. In what context we are trying to figure it out the stability of Ganga if it is diverse?
Well, you see, by stability, I mean, pollution. Okay, as it moves, for example, Gangotri is the starting point of Ganga. Okay, so it already has a natural community there. At that, it's in the cold region, but there is a natural community there. As it passes through different zones, all right, because of the man-made activities, because of the pollutants, because of the discharge from various industries which are entering into Ganga. Okay, the kind of diversity which you are going to see at upward points is not going to be there at the downward points. Because of pollution, many of the microbes have been completely wiped out. So the diversity, and if it is very rich in domestic sewage, for example, you find that the diversity has already gone down. low, because there were many microbes which were originally growing at 23 degrees temperature under those kind of nutrient condition. There is, so there is a certain microbial structure, community structure. But as more and more pollutants enter into the river, the community changes and the diversity of that community also changes. So therefore, looking at simply microbial diversity, the question is, can it also serve as an index of the purity of the water?
Okay,
So
ma'am.
that's a big area. Okay,
Understood.
so is that clear as to what we mean by that?
Yes, yes, ma'am.
Okay, fine. So I'm going to stop here and then we will come
Um,
back.